actually time.
Yeah.
Good job. Alright. We have for each one. Actually,
Oh, I did you have to get that.
Okay, so like I said, this is a pretty simple question. What's asking you for the complementary strand? It's asking you for the base pairs that would go across from it, right? So all you have to do is use your base pairing rules. A pairs with, B and T pairs with, A and C pairs with, C and D pairs with, and C pairs with, and A pairs with, and G pairs with, and C pairs with. Okay. So that's probably how you would write it. Now, you kind of have to remember, though, that these strands are running opposite of each other. So technically, if this runs from 5' to 3', this is going to be 5' to 3' here, right? So we mentioned that. Difference in directionality. So today we're going to start talking about DNA replication, and that'll take maybe about half the period, and then I'm going to give you the rest of the time to finish up that modeling activity from yesterday. So, and you'll have some time then to update your study guide a little bit if you haven't done that yet. So, I would go ahead and get your notes out. If you haven't already. But if you have that DNA model, you can just put that away for now. I'll give you time at the end of class, but please don't work on it right now. So this is going to be the next section, section 9.3. So before we get into it too far, we're going to ask a couple big, important, overarching questions for this section on DNA replication, which is what happens during DNA replication, and when by we I mean cells, when would cells have to do that? Okay, take a couple seconds to talk about that in your group. What do you think?
They taste no, one strand. Excessive constriction of the pupil of the eye.
All right. Okay, let's start real simple here. I heard a lot of good things out there. So, the basic idea is that when we replicate our DNA, we're doing what? Yeah, well, just replicating DNA means we are copying, not yet. We're doing it so we can make two cells, but replicating just means we're copying the DNA, right? So this process we're going to look at over the next few days is just how do we go from one double-stranded DNA molecule to two. Okay. So how do we copy the DNA? We do that in order to make new cells because each cell does need a whole copy of the DNA, right? So if you look in any of your cells, you're going to have the same, you should have pretty close to the same 20,000 genes in each one of your cells, right? 46 whole chromosomes in every single one of your body cells. Okay. So that's actually the next unit we're going to talk about after this is cell division. So we're going to talk about the DNA replication mechanism over the next couple days, and kind of again keep in mind that we do that process in order to pass on that DNA to the next cell. Right? So jot something down about that in your notes. So even if it seems simple, write it down real quick. Well, what do we do when we replicate DNA? Copy it. Why do we copy it?
Yeah, like a lot.
So we have a copy to give to a new cell. So we do it before cell division so that when the cell divides, each cell can have a copy. So this is really going to be another history lesson today. So after Watson and Crick came up with the final model for the DNA molecule, we can now start to turn to the question of how does DNA copy itself? Because we know DNA gets passed on to new cells, so how does that happen? So at the time, there were three competing ideas or three competing hypotheses for models of how DNA gets copied: conservative, semi-conservative, and dispersive. Take a couple seconds to compare those three models with your group and kind of talk about why they are called that. So why do you think they're called conservative, semi?
You see, it takes like things that you want to say. Nothing. There's like only like one. From But I don't think it's semi. It's like when you split it in half. Takes a little bit. Actually, no, I know.
All right, so let's take a look at these. First one's called conservative. So conservative means to keep the same, and so in this case, our original DNA strand stays together, right? So it's conserved throughout the rounds of replication. So we're somehow just making a new identical molecule that's separate from the old one. Right? And when that divides, that old one is still kept. Okay, so that's the conservative model. In the semi-conservative model, what's different about that? Half of the original DNA strand is kept intact, and we're just adding a new strand to the other side. Okay, so semi means half, right? We have a semicircle, it's half of a circle. So semi-conservative, half of the original DNA molecule is kept intact, the other half is new. Right? Dispersive just looks like random pieces are old and random pieces are new, right? So there's not really an order per se. But every strand of DNA contains some old and some new. So it's dispersed. The old and new is dispersed on each one of the strands. Right? Questions on that so far? Yeah. Yeah, so in semi-conservative, half of the molecule, one strand is kept intact, right? And so the other half is one half goes into one molecule, the other half goes into the other molecule. All right, here's something else to think about. how do you think we would the test for this model, or what would we need to be able to do to test for which model works? Stop on that in your group real quick. Alright, so any ideas on what we might have to do in order to test for this model? Maybe somebody hasn't contributed yet. Yeah, so you're thinking about the Hershey and Chase experiment a little bit, right? Yeah. So they did use isotopes, but not to test the radioactivity. So when you use different isotopes, what else are you changing besides whether it's radioactive or not? The number of neutrons, which affects the... No, not the number of electrons. That's the same for isotopes. Not the number of protons either. That's the same for isotopes. The mass, yeah. So the DNA will have different masses depending on how much new DNA it has versus how much old DNA. So that's what they did in this experiment. So back in 1958, so this experiment showed that DNA replicates via semi-conservative replication. The scientists that did this experiment were Messelson and Stahl. And so what they did is they used different isotopes of nitrogen to differentiate between the original DNA strands and the new ones. So when they started growing, they used E. coli bacteria for this because they replicate, they divide pretty quickly. They labeled the parent nucleotides or the original DNA with a heavy nitrogen isotope, say nitrogen 15. So they started growing cells in this heavy nitrogen 15. So eventually, over time, all of the DNA in the bacterial sample is made up of this heavy nitrogen. So they switched over to growing the bacteria in regular, more regular situations. So the more common isotope of nitrogen is nitrogen 14. So the parent DNA had the heavy nitrogen. Any new DNA that gets added is going to have the lighter nitrogen. Okay. So let's take a look. So here's the diagram down here. In the first round, we're just growing bacteria in that heavy nitrogen. So all the parent DNA is going to have heavy nitrogen in it. They transferred the bacteria to the nitrogen 14. And then after about 20 minutes, which is about how much time it takes for the bacteria to divide, they centrifuge the sample. So again, what does centrifuging do? Separates separates things that are heavier or lighter. So any DNA that has the heavier nitrogen is going to be lower. Any DNA that has the lighter nitrogen is going to be higher. So you'll see bands. So nitrogen, if it was all nitrogen 15, so 15N, 15N, it's going to be the lowest. If it's half and half, it's going to be in the middle. And if it's the lighter, it's going to be the top. Yeah. Okay, so there's 3 different bands where our DNA is going to fall in. Okay, depending on how much of each nitrogen isotope it has. You with me so far? Okay. So, if we're looking at the conservative model, after the first round of replication, what band would we expect to see? Which of those 3 bands? Yeah. The very top and the very bottom, because the DNA is going to be all old or all new. After the second round of division, what would you expect to see in that test tube? Still all old or all new. So you might have a little bit larger band of the all new, okay, but you're still going to have those 2 distinct bands. Right? So that's what the conservative model would look like if that's what's happening. Go ahead and talk to your group members. What would your predictions be for the semi-conservative and for dispersion?
So for the second, so surely they have to be high. For the clear,
Okay. So, semi-conservative, after 1 round, what band or bands would you expect to see? You'd have how many bands? 1. And it would be somewhere in the middle. It would be right in the middle of this, right? Because half of the strand is old, is made out of the heavy nitrogen, half of the strand is new, made out of lead. So 14, 15. After the second round of replication, how many different bands would you have? 2. Okay, because half of the DNA should be 14, 15, and the other half should be 14, 14, right? So you'll have the 14, 15 band, and you'll have the 14, 14 band. That makes sense? No? Okay. Where am I losing you? Okay. So if you look at the second round of replication, half of the DNA should be completely new. So that's all going to be made up of nitrogen, of the lighter nitrogen. So that's going to be 1 band, the highest band. Okay. The other half of the molecules are going to be half 15, half 14, half gold, half new. So they're going to be, their mass is going to be in the middle, 14, 15. For dispersive, how many different bands would we have after the first round of DNA replication? Just 1, because each one of those molecules is an even mixture of old and new. Okay, so it would look just the same in the first round as the semi-conservative, right? So that's why we have to do this second round to determine. So in the second round, how many different bands should we have? We should have just 1, Because again, each one of these is the same combination of old and new DNA, right? And so it might be a little bit higher up because as we go, we're getting more newer DNA versus old, but we're just going to have one band. So it'd be, some combination of 14, 15. Okay. So, I think there's a lot of animation here. Here's the actual data. So here's the original DNA before any replication. So we're starting with DNA that's all nitrogen 15. Here's after the first round of replication. Here's after the second round of replication. Okay, go ahead and analyze that data in your group. What type of replication happened and why? The original DNA has what in it?
We went in these separate directions.
In the second round of division, we got rid of the band. So half of it still has
So those things could walk away. They would the amount of
some of the DNA. Wait, hold on a second. Thank you. Okay, so hopefully you determined that this tells us that DNA replicates using what model? It's semi-conservative. Okay, so if you look at these bands, so we start with original DNA, all N15. After the first round of division, these molecules are half old and half new. So we end up with that band that's in between, right? Half old, half new. When we go through the second round of division, half of the molecules still contain that original DNA in a new strand, 14, 15. The other two molecules are going to use the new strand from the first round of division, and so they're just going to be new and new. Okay, N14, N14. Yay, nay.
Yay.
So. Okay, so this is the very simplified version of DNA replication. We're going to get into a little bit more complicated, so we're going to dig into a little bit more tomorrow. But when we copy, if you just take a look at the diagram here, so here's a DNA molecule. The first thing we do is to break it apart. Okay, so what are we unzipping here? Yeah, what bonds are holding those together? So we break the hydrogen bonds between the two strands. Then we add in the new nucleotide, and we know what to add because of the base pairing rules, right? So we know if we have an A on one side, we're going to add a T. If we have a C, we have an E. The opposite strand is going to be the opposite code, but when we copy it, we're going to still be adding the base pairs. And so as a result, we get two identical molecules. So they have all the same sequences, right? But it's half of the original DNA and half new, half original. So each side of that DNA molecule is acting as a template for a new strand. So that's the very, very simplified version of DNA replication. We'll talk more details tomorrow, but I want to give you some time to finish up that assignment from yesterday. So I'll take some time to do that. You need to take pictures of anything that you wrote or drew and put that into the Canvas assignment. You're supposed to exchange your DNA molecule with somebody else to see if they can figure out who your person was.